Welcome to this latest video where I will present the innovative result of Dr. Pankaj Singla, which was a result of his Marie Curie fellowship, and he used a double imprinting approach in order to achieve specific and selective drug delivery to breast cancer cells. And this was used to treat estrogen positive breast cancer, and the majority of the breast cancers are estrogen positive, but because resistance can occur, there's still a very critical need to develop novel treatments, and actually the loss of this estrogen receptor is associated with the resistance developing. So a lot of our research is focused around replacing antibodies. So antibodies are what we naturally have in our body to fight off foreign invaders, and that's because they work according to a key and lock mechanism. That's why they're so good at recognizing specific bioanalytes. So we've seen there's a growing market, particularly in terms of therapeutics and drug delivery, where antibodies are being used, mainly in cancer treatment. However, the problems with antibodies is that they're very expensive and they're not very robust either. So it means that a lot of these therapies are not generally accessible, even though they exist, simply because of the cost. So what we're trying to do here is make plastic antibodies. So we make alternatives towards these antibodies. But I've got to say, even though we claim that these are plastic antibodies, they're very different in structure. But what they are is they're very low cost materials which are very robust, so they're not very sensitive to changes in pH, which can occur, for instance, in tumors. They're not very sensitive to changes in temperature, which means we can autoclave the materials, so we can sterilize them. And we can easily produce them in bulk, which is very important. And we can produce them without much variation between them, which can be another problem that's related to antibodies, which sometimes can also cause some allergic reactions. Well, for drug delivery, you can't just use what we call like the traditional approach, where we actually have selectivity for only one analyte. If you want to achieve drug delivery, we need to have the selectivity for two particular analytes. So one is, if you look at the protein that's overexpressed in cancer cells or something that's unique to the cancer cell. And the second is a drug compound that we're actually going to deliver towards the cell. So until date, there was only one paper that worked according to this. And in this paper, they looked at EGFR, which is another protein which is overexpressed in a number of cancers, which is present on the membranes. So they imprinted with that particular protein and with a drug compound, in this case doxorubicin. So it's a chemotherapeutic agent. So what happened is that it had both, this MIP was very specific at recognizing the receptor which is overexpressed at the surface of the cancer cell, and then once it bound to it, it would slowly release by diffusion this drug. So this would be very interesting for drug delivery. However, if you want the drug to be effective, it's key for it to enter the nucleus. So this is where this particular part is so unique, because we developed a system where actually the drug will enter the nucleus, which means the efficacy is much higher. And that was something that hadn't been described with these double imprinted nanoparticles. And actually the fact that there's only one other paper to date shows that this is a very innovative process. And part of the reason probably for that is that the synthesis conditions are very specific. So imagine when you do the synthesis, you have made make sure that you can solubilize both the drug and the protein. So the protein traditionally is soluble in water, whereas the drugs are not always soluble in water because they tend to be hydrophobic, so that can be very hard to balance. And you need to make sure that you balance the affinity for both the compounds in order for this to work. We looked at double imprinting with this estrogen receptor, and then with a traditional chemotherapeutic drug, which is doxorubicin, and applied this approach in order to treat breast cancer more effectively, whilst also minimizing the side effects. So what's different in a double imprinting is that you have two analytes. The primary analyte is the protein or an epitope of a protein that you're interested in. The second one is a drug compound. So the primary analyte is attached to the glass bead. The secondary analyte is present in the monomer mixture, but there's no covalent attachment like you've got for the primary analyte. It does mean there's some kind of preference for the secondary analyte and some imprinting going on. But the binding is less strong, and it means that the delivery of the secondary analyte, the drug, is governed by diffusion, so there's less control over it. So key here is to use epitope imprinting. For this solid phase approach, we need around 1 milligram of the template, and as we know, proteins are just simply way too expensive to use in this case. So Either you can use some computational modeling for this. In this case, because we knew we were working with estrogen receptor, of which you have the PDB files, and we also know that there's some antibodies that bind to very specific places on the estrogen receptor. The antibody binding domain, we obviously need something to attach it to the solid bead. So commonly people would add a cysteine to it, and you can just order it from a company that will produce it for you in the quantity that you need. The advantage is as soon as it is on the solid phase, so as soon as it's attached to the bead, you can recycle it. So even though it's still expensive to order something which is like a 10 amino acid sequence, we can use it for quite a long period of time and can produce quite a lot of material from it. We produce different batches of nanoMIPs, one that were targeting the estrogen receptor, ones that were targeting estrogen receptor and had doxorubicin within the polymerization mixture. And then we also looked at embedding a fluorescent moiety, in this case fluorescein, in order to probe the interactions between our nanoparticles and the target that we're looking at. So the first thing we need to have a look at is the affinity they have for the particular protein that we're working with. So this can be achieved with a variety of different techniques. So shown here, you can use some fluorescence studies, which we can do because we have a fluorescent moiety in a material, so we have fluorescein in there. We can use ITC, which is also a commonly used technique. Downside is that you need a lot of materials. So normally we stick to surface plasmon resonance, which is an established technique. So you would attach your molecular imprinted nanoparticles to a surface, and then you would flow over certain concentrations of, first of all, your epitope to show that we can achieve binding to the epitope, second, the protein, and then third, we would look at some selectivity by either looking at different proteins or scrambled versions of the epitope to show that we have very specific binding towards only that sequence. Now here you can see how this process worked, and we were able to achieve binding affinities, which we normally represent as KD values, that were in the nanomolar range, which are very comparable to commercial antibodies. So that would be a good indication that we actually have the affinity that we need for the protein. And what you can see here is that it works not just for the epitope, but also for the protein itself, for the full sequence. So using the epitope approach was successful in really having a compound with a very good affinity for the overall protein. But this can only work with the protein itself. It doesn't work with cell lines, even though there are some SPR systems that can work with cell lines nowadays. So in order to test this, we worked with some two specific cell lines. The first of all was estrogen positive. So MCF7 is a model cell line for estrogen positive breast cancers. And then we worked with another cell line. So this was a mimic of triple negative breast cancer. So normally it shouldn't be estrogen positive, but it's seen because estrogen receptor is overexpressed in breast cancers, there was still some residual estrogen receptor present within those cells. So we used flow cytometry, and we could do this again because of this fluorescent moiety. And what we could see here is that there was much more binding to this MCF7 cell line, so the positive cell line versus the negative cell line. The negative cell line still had some binding, but that was expected, first of all, because there was some residual estrogen receptor. And you also have to imagine that even commercial antibodies or commercial drugs, they do have these side effects, which means that there is always a level of non-selective binding. Obviously, we try to minimize this, but you can't rule it out completely, unfortunately. So we show now that this works on a 2D cell system. The next step would be to actually test the drug efficacy. So this is normally tested with some biochemical assays. So we can use something like an Alamo Blue assay. In this particular case, we use an MTT assay. And we tested this for a number of different nanoMIPs, so a control nanoMIP, one where we used fluorescein itself, one which was actually imprinted for the receptor with fluorescein, and then also one where we had the whole thing, which was both doxorubicin imprinted for the estrogen receptor and having a fluorescein moiety. And the key thing is we don't want a burst release. We don't want this to happen just instantly, because that would lead to more side effects. So we tested this over time to see over 3 different days whether there was a temporal release that we showed. Now, here we worked again with the two cell lines, and what we could see that it was much more effective in killing off the MCF7 cells due to binding to this estrogen receptor versus the control cell line. So that was a good sign. Now we know that we have affinity for the estrogen receptor. We can effectively kill off the cells, and the next step would be we see that the efficacy is quite high is this actually the case because of having this nuclear delivery? Because as I said before, because in previous papers this wasn't investigated. So we know the estrogen receptor is present on the cell membrane, but also in the nucleus, and the mechanism that is postulated to work with via endocytosis is that these receptors move into the nucleus. So would our particles actually move into the nucleus according to the same process? Well, we can't really investigate the pathway with what we did, but we can use confocal imaging to show that the nanoparticles actually went into the nucleus. And if you do this over time, and this happens relatively quickly, you can see that initially they're only present on the cell membrane, and then after about 30 minutes they move into the nucleus. And they take the drugs with them, so that also means that we get this nuclear drug delivery, which makes this research so unique, which leads to higher efficacy. And this would really mean a breakthrough, because before you were only able to target membrane receptors, which leads to very poor drug efficacy and high amount of side effects. Now, the way you can see this is the nanoMIP was obviously had an embedded fluorescein probe, so it was in green. Then we stained the cell membrane, and then we stained the nucleus with TAPI in blue. And you can see over time that there is really a push of these particles into the cell nucleus, which makes this research so unique, which leads to higher cell death. And you can see this cell death by the deformation of the cells as we go along. However, these are simple 2D cell systems. This doesn't really mean anything in terms of a patient-like tumor microenvironment, which is incredibly complex. So in order to move towards clinical trials, what we wanted to have a look at is, first of all, are these systems biocompatible? And the second, how would it work in clinical practice? Can we try to predict this? Now, the first thing in terms of biocompatibility, this is not very well understood for these molecular imprinted nanoparticles. There's simply no clinical trials out there. Actually, even if you look at animal models, the data is quite limited. So here I've shown like a paper where they looked at some of this. So they looked at the biodistribution. So if you use like a model system for a nanoMIP with a very typical size, typical charge, they could see that it ended up everywhere in the tissue. It was cleared in urine after a short amount of time. And what they also found out is that it actually crossed the blood-brain barrier, which might present some opportunities if you look at neurological diseases, but this can also be a downside if you look at, for instance, how your immune system responds to this. So some mixed results here, plus you have to imagine that each nanoMIP is unique. So you would have to have like a standard protocol in place to actually test this impact. Now, what they found out in this paper, and this is actually done for about a week, so people haven't really looked at long-term studies. They did find there was some toxicity, which was very dependent on the dose, which is logical. You can only have a very specific dose of a drug, but very important, the administration method. So they could see that if you take this orally, it was much more toxic, which they evaluate by looking at concentrations of CRP and interleukins, compared to when this was an intravenous injection. So this is something to bear in mind. As I mentioned before, it's very highly dependent on the size of the particle that we've got. Also, the mechanics will have some impact, whether this particle is hard or soft, the charge, and each of the monomers, so the chemicals. So even things like your initiator, you need to carefully consider each of the components that you're using. So the FDA Modernization Act shows that we can work with other systems. So for instance, we can use organ-on-chips or 3D models, as we did in this paper, before having to go on to clinical practice. And even computational modeling has a role to play here. So we were working with a group at UCL of Dr. Value, where we showed that we can use very sophisticated polymer systems as these 3D models, because they can mimic the porosity, they can mimic the chemical compositions by adding in proteins in there, and they can mimic the mechanical strength. So we have shown here, again using confocal imaging and MTT assays, that first of all we can visualize these nanoparticles into these systems, we can culture the cells for longer periods of time, and then we can again evaluate the efficacy, where you can show it's more effective at targeting the breast cancer cells that are positive for estrogen receptor versus the control cell lines. But this is only really a start, and we would really need to look at more patient-like environments or patient-derived cell lines in order to show that this works. The disadvantage of these patient-derived cell lines is that they normally don't survive for very long, so it's very difficult to get this temporal response. So in summary, what have we shown? These nanoMIPs can rival the affinity of traditional antibodies. So they have a place in cancer treatment and potentially a new class of drugs by being very specific at targeting cells. So they can lead to higher efficacy and also minimizing side effects. So in order to move this forward, what do we need to have a look at? So I've mentioned these patient-derived cell lines, but people are still consider, despite this FDA Modernization Act, animal models the gold standards. So we probably would still be looking at establishing a protocol where we look at the particular composition that we used, how would this work in xenograft models. So looking at the clearance, the biodistribution, and also what dose that we need in order to use for our treatment. So thanks for watching. So hopefully this will give you like a teaser for the paper. Do have a look at the paper for actually more of the scientific content, which you can find via the link in the description.